They're very tasty fish and a potentially commercially viable species. However, this is a protected area, and they live at depths that make it a little less economical to fish for. And so this is great to explore their habitats so that they can be protected from overfishing, because they are definitely top predators.
All right, so
yeah, after we get to this next waypoint-ish, we can start turning.
And Chris Ma, our echinoderm expert, has let us know that Ophidiaster. And thank you, Megan, for relaying that information. I'm sorry if I didn't give you credit for the previous relays. I thought it was Tara Luke. So Ophidiaster, we have not seen that before.
That dogfish again.
Yeah.
And the wreckfish is in the genus Polyprion, and that means, in Greek, poly means many, and prion means saw, and that's referring to their spiny fins.
Video, were you talking to me? Ah, copy.
I think we should take a moment to introduce ourselves.
Copy.
We have on the back row here, we have Derek Sowers, who is the mapping lead. He doesn't have his headphones on, and he doesn't want to talk, so he's just waving.
He's busy.
He's busy. And there he is. And Derek has been up many hours. Many of these dives, we have to map the previous night, pick a site, and then he spends the day working with the prep for the next day, too, especially now that we're in the shallower waters. There's not much time to sleep.
To your left as well.
Maybe he'll say hello.
Hi, everybody.
So, Derek, what time did you have to get up today?
Well, we were all up at about 5.
Yeah, well, some of us got to go back to bed. You did not.
So one way is 100.
Yeah, we want to look more.
But it paid off. I'm glad we were able to get here.
Yeah,
Very interesting.
yeah, we had not originally planned to come here. We were in search of. You want to talk a little bit about what we tried to find yesterday or last night?
Sure. Yeah, we didn't have a lot to work with, but we needed a stop somewhere between yesterday's dive site and tomorrow's, and it had to be about this distance where we are now. So the best data that we had to work with was really the satellite-derived bathymetry for the region, which is very coarse resolution on the order of about 1 kilometer. And it looked like there was some relief that we could explore and map in high resolution and see what was there. And we did that overnight, and it ended up being just a very gentle slope, essentially flat seafloor. No real relief features for us to dive on. So we knew this was nearby, and it was definitely a definite target that we could get the vehicle down to. So we had to make some last-minute changes. But that's the nature of exploration. You never know what you're going to find until you go out there and map in high resolution and see what's actually there. So we'll be mapping again tonight, and who knows? We'll be doing some shipwreck surveys actually the next 2 evenings. So with any luck, we'll be able to hopefully find some promising targets to dive on later.
And all this mapping data becomes available to anybody, and other scientists will use it for lots of other studies in the future as they plan their dive sites.
And my students will be using it as well in our seafloor mapping program. They use a lot of the Okeanos Explorer data, which is high quality. You know that it was planned well and conducted well, and it's very clean data.
Do you just want to split it, or
and great training data as well as to use for small research projects.
Say again?
Thanks, Leslie. Yeah, and we love to see your students, what the amazing things they do with it, and just to know that they're both learning about how to work with this type of data, do their own creative analysis, and just really taking advantage of this publicly available great data set that's out there. It's really gratifying for us that collect it to see it get used and see people really digging through it. There's a lot of information to mine through that we often don't have time to go back and do. So
yeah, we get to look at it in a lot more detail than you ever have a chance to because you're on to the next day.
Yeah. Yep.
We forget that. Yeah.
And as a reminder, we do really put a lot of effort into trying to not be redundant with our mapping. So we're always looking at mapping that was done previously through any type of cruise and try to always get new seafloor if possible.
And this area that we've covered during this cruise is huge if you look at it on any of the maps of the overall cruise plan. And so it takes a long time to collect this good high quality data over this whole area. So we've been trying to contribute to that every night that we've been out here.
Yeah, the advantages of having available maps really played a significant role a few days ago, where we had hoped to, we were using recently collected data on the last leg of this expedition, the mapping cruise, in an area called Savannah Banks, so we had a target dive, and it turned out when we were to go there, the Gulf Stream's axis was right over where we wanted to dive, so we had to plan for a different dive. It was a little farther east, and then had to go a little farther east. So we were taking advantage of those very new maps and finally found a spot that was diveable just outside the edge of the Gulf Stream, and it turned out to be a very surprising dive, a great dive. We expected it to be great, but we thought it might be a rocky ridge, and it turned out to be a knife-edged, very eroded, the edges were very eroded, but it was a coral mound, and it was much farther east than I certainly would have expected to find a coral mound.
Yeah, that was really surprising.
So, yeah, that was all in the nature of exploration. We expected one thing and found something very different, and so now that kind of has shed light on what a lot of the features on the seafloor that don't really look like rocky outcrops, they may well be these coral mounds. So it could be a lot more habitat out there than we realize, and we won't know until we keep mapping in very high resolution and keep visiting and ground truthing some of these areas.
So I'll push out a little bit, and then we could start that move.
Yeah, sounds good. I have you at 60.
Okay.
Camera.
Oh, and if getting back to the introductions,
that's right.
That's what we were starting to do. Lots of great conversation in between. I'm Cheryl Morrison from the U.S. Geological Survey Leetown Science Center.
And I'm, oh, go ahead.
Oh, I'm the biology science lead.
And I'm Leslie Sauter from the College of Charleston Geology Department and the BEAMS program, which is our seafloor mapping program that we were just discussing. And I'm the geology science lead. And Cheryl, what is your background? You're at the U.S. Geological Survey, but you're a biologist, so that might confuse some of our listeners.
Right.
And viewers.
It would make more sense if you were from the USGS and I was from the College of Charleston, but it is the way it is. And so USGS is the science branch of the Department of the Interior.
About 11 of those.
The Fish and Wildlife Service is part of Interior, as well as the Bureau of Ocean Energy Management,
like 7.
and the Park Service, National Park Service. And so USGS does science that will help any of those other agencies manage their resources. And so we have been working with BOEM for well over a decade now.
Let me know
and
when you're
we
ready to
help
get, ship moving.
add expertise
think
that
we're
explores
ready.
these types
Yeah,
of
I did about
habitats
50.
to characterize them
looking at,
fully
yeah.
from the microbes
Still
up
facing
to the
125,
biogeochemistry
looks
and
Looks pretty
tell them
go a
as
little
much as
more
we
east
can
and forward.
about the species that are living there and their
I don't know if you wanna
distributions.
curve it or
And
just do
BOEM uses this
let's
information
do 135
for
at the
areas of
beginning,
avoidance when
and
they're
then we can
leasing
copy.
parts of
Yeah,
the
sound
seafloor
good?
for
sounds
Okay.
great.
extraction
Ready
of
to move now?
resources such as
35.
oil and gas, or even
Hey, we're ready for
wind
a ship
farms,
move.
which are something that
30
will
meters
also
at
be
135.
potentially
0.29.
impacting these areas. But with the correct information, we can make sure that we have the resources
copy.
available to the public that are needed,
Is that
but
3 or 4
still
sea urchins
allow
there on the right?
these really sensitive habitats to exist. And so we need to plan what our human activities in this ocean
like 6
realm
of them.
so that we meet the balance between what people need and what has taken hundreds of years to grow. And
Thousands.
thousands of years,
Thousands.
seriously. Yeah, hundreds is a really poor estimate. So thousands of years. Some of
Take
the
a snap
black
from
corals
here.
that we've seen on previous
Okay.
dives are some of the oldest living known
ahead.
organisms and can be over 4,000 years old. So, oh wow, look at that, a whole group of pencil urchins.
Oh, they are feeding on some sargassum, it looks
Yeah.
like. How interesting.
And a lot of urchins are herbivores, but there aren't very many plants down here. So when seaweed from the surface finally reaches the seafloor, I guess the dinner bell rings, and
I guess.
that's great.
I was going to say we haven't seen very much of it, but that's probably because all these organisms are waiting to feed on it, and it's already gone. Hard to say, but there certainly is a plethora of these brittle stars, the pink stars, and
come back in video.
many
Copy
of
that.
these pencil urchins. Everybody's
say
coming
in
to
or
the
out?
dinner bell.
Yeah, zoom in. Go wide.
Okay.
They're moving in.
You can lose this kid, so.
And so as part of our work with the Bureau of Ocean and Energy Management, we have a project that is a longer-term multi-agency project called Deep Search, where we're going to be characterizing a lot of the
Check.
communities in this very region, and including the canyons and the seeps where we're going to be this coming week, to allow the best science possible for BOEM if it does come to the point where we are going to be leasing parts of the Atlantic Ocean out here for oil and gas development. So this data will be used directly, very quickly.
Yes, I wouldn't recommend that the base of one of those very large wind farm, the wind turbines. I was going to say the windmill, but that's not an accurate description. The base of one of those here, with all that force on the wind, it wouldn't be very stable, I'm quite sure, and boy, would it damage some habitat. Of course, that won't happen here. This is a marine protected area, but we're finding more and more of these mounds as we explore.
Yeah, it looks like we're getting more of the Lophelia, more live colonies, so the bright white colonies that you see. And basically the brown skeleton that you see all over the mound is dead skeleton from that species. And it likely sticks around for a very long time, and we've been collecting parts of this coral rubble to do some aging work on it.
And by skeleton, we really mean an exoskeleton, not the type of skeleton that vertebrates have. And the polyps live within the exoskeleton.
And I should add about that deep search project, that is, the agencies involved are USGS and BOEM, as I was speaking about, but also NOAA's OER program. So the same program that is running this vessel is also our partner in the deep search project. This kind of work out here is expensive, but the information needs are similar across agencies. So when we partner and combined resources, we get a lot of information out of the time at sea and back in the labs when we process data.
See the bunch of yellow stuff upper right? Just went off screen.
And is this a sponge up in the
Where
upper
is it?
center of
Those
screen?
yellow dots upper right.
Might want to take a quick zoom on that.
On
think
those
it's-
yellow sponges, Watchley?
No, it's just going off the left corner.
Okay. I'm coming around.
I think it's the same thing we've been looking at, but we haven't done a quick zoom in a while. There aren't very many of these.
Let me know when I line up with it, Watchley, please.
I think it's one at the top.
I think it's that one on the top.
The sponge copy.
Another pencil urchin in the at about 5 o'clock.
And it looks like another reef codling in the upper right, trying to hide from us. Lamanema
Asia's
melanorum,
clear.
Okay.
also called the coral
Copy
hake.
that.
Ready for zoom.
Stand by. Okay.
There are so many of these round sponges and so many outer appearances. Some of them are prickly like this with sediment. Some of them are smooth. And actually it looks like there are feather duster worms growing on this one. That's interesting. Wonder if the sponge is still alive.
Perhaps a scavenging worm too, right in the middle upper part of the screen at 12 o'clock.
Max soon.
Polychaete worm.
Video's clear.
Did you get what you needed there, Watchley?
Yes, thanks. That's
Okay.
That's
All
great.
right. Full wide video.
You can see those spicules
Pilot, I have
end
to,
up in our
sit
sediments
down
and on our
the camera.
samples and in
Copy
our
that.
skin.
So
Yeah.
Stop moving, so
All right. I'll
you're in
push
no
forward.
rush.
Oh, wow.
Hello, wreckfish.
Lasers on again.
They are certainly not camera shy.
They are not. Oh, this is great. His buddy said, "No, you go check it out."
Go scare them away. Tell them to go home.
Got any squid?
He does have a little parasite on
Yeah.
his side there.
Okay. Full wide video.
You are,
Okay.
except for the skids.
All right. Thank you.
Go ahead, Rich.
This has been a day of fishes.
Copy
Lots of
that.
new species
Should
that we
hold
haven't
off on any
seen
moves, or
on previous dives.
Copy
all
that.
of which
Thanks.
are
Thanks for letting me know.
not too much of a surprise in this habitat and at this depth and location.
I saw it sorghum in the water this morning.
But it's important to note that these
Oh, yeah.
fishes are using this
And it clogged it up.
coral habitat.
Does that. Pilots, you guys want to just discuss an emergency
I guess those are just branches on the right.
situation in case? I was thinking we would turn pilot directly to port and line up and push out. The ship is going to move that
So
direction, so
fuzzy
maybe we
thing
want to change that.
Diagonally up from lasers.
Because
What's
the ship,
doing?
it's going directly this way.
So you would push to the northwest?
Oh, that's. I think we should change that.
Okay. So what are you thinking now?
To the west.
Go ahead, Bridge. Copy that, thanks. So, yeah, that's what I would think I would do now. Because what the ship would do would blow us back this way. It would blow us to the northeast,
Okay.
the current will, and the wind. So if the ship loses power, we're definitely going this way, northeast.
So if that's the case,
You could just turn.
Sirius
Right
could just
now I
turn.
go to starboard, right? I mean, it put a turn in,
That would
maybe
put a
quicker.
0.5 turn, but then Sirius could turn as well.
The quickest would be Sirius
And we're at 400 meters now.
Yes,
faces the track the ship would take, and D2 goes starboard.
that would put a
Oh, yeah.
turn in there. Is that correct?
And it'd have it, yeah.
Okay.
So that'd be the
That
We've
quickest.
been hovering between 410 meters and 400 for a long time, so we've been exploring this kind of saddle that leads to the crest of this mound. And as we move up
But
towards that crest, I bet you we're going to see more and more of the white live cover of
What is Bela
Lophelia
right now?
on the mound.
it's still.
Pilot, can we do a quick zoom of the sponge we're passing? It's right in the center to the left.
What's that, Watchley?
The sponge
Okay, will you be ready for
to
ship move when we are?
left of your lasers
Okay.
now.
Okay.
Baylet's kind of
Yellowish.
northeast because that's the way that everything will go.
Looks
We
a
can
little
start
more
changing it to
elongate than the
northwest, though, because
barrel-shaped
now
ones
that we're
we've
gonna be going
seen.
upslope, that'll be the quickest way off the slope.
Yeah. That sounds good.
Bridge, RV Nav. Hey, we'd like to
Would you like a
update
zoom
our
on this,
bailout plan
Watchley?
as well.
Yeah, just a quick one, thanks.
Okay.
Okay, we'd like to change the bailout plan to northwest. We know it's going to take a little heading change, but that'll be better for the bottom.
I'm working on a zoom
Apology.
requested by Watchley right now video. All right, you want to come in?
Excellent.
Oh,
Thank you
great.
very much, Bridge. Hey, pilots,
It
are you
looks
ready
like
for
we have
ship
a
move as
couple
well?
more of the nudibranchs that we
Yeah,
saw earlier.
Yep.
Pilot 3-5?
Okay.
Or no, maybe that's an anemone.
I think it's the anemone
sorry.
because there's one on the
I was
left.
looking at my
Looks the
camera
same.
angle. Whatever.
Okay.
But yeah, they do look really
They
similar,
have the
don't
same
they?
white
140,
Uh-huh.
there's
little
not much.
tentacles.
Copy, 140.
Bridge, RV Nav.
And
We'd
there's
also like to
something
put a ship
living in the
move.
spines. It's a brittle star living among the, not
That's
spines,
correct.
but
30
the
meters at
spicules.
140, 0.2
Good
knots.
grief. It's an interesting habitat.
thank you.
Yeah. Well, it's probably fairly safe there.
Yeah.
Video's clear.
Okay.
That's great, pilot.
Hawaii.
Thanks. And to the left, there might be something.
Thanks, Bridge.
Yeah, I think there's. I'm not sure
Upper left.
what
of
Roland
lasers,
was referring to.
orange and white striped anemone.
Yeah, I think that's a Venus flytrap. But yeah, let's take a quick zoom. We probably haven't done that in a bit. It does have different coloration than the other bright orange one on the lower left.
What's the one above the lasers?
Yep, I'm just setting up. Okay.
Kill lasers.
I'm not sure. Well, it's a beautiful one.
Zoom past.
Yeah, that is. I guess it is a Venus flytrap. It looks very different. It might be different species.
Yeah.
Good eyes, Roland. Roland is our video lead at the present.
Video's clear.
Okay, full wide.
You can see this anemone is facing right into the current.
And pilot, can we just lower down right below that? There's a cluster of anemones. Or at least that's what we think they are. See what they're clustering around.
Definitely looks like a cluster of anemones.
We wouldn't call that a school of anemones, would we? And some more of the hydrozoan. Is that something we might want to collect, try to collect?
We do want to try to collect it. I don't know if that's the most convenient spot.
Video's clear.
Yeah, the ship's still moving, right?
Yeah.
Yeah, it'd be co-pilot. How do you think I could grab that? I think if I grabbed it with the claws, it would just kind of fall apart.
Mm-hmm.
That's
Yeah,
what's
just
happened
like,
previously. It falls apart.
It's going to be an attempt.
It's
I can
so
stop the ship
small.
if it is.
Yeah, watch it. I don't know. I think the same thing would happen like last time. It would just crumble if we went with the claws.
Yeah.
And each individual item's too small to grab independently.
We're probably going to leave these guys alone.
What's that? Watch one?
Watch
They're
one's
pretty
called
small.
Pilot.
Okay, so should we move on then, watch lead, or should we?
Great.
Okay, all right, full wide video.
And
It is interesting, though, that
as you
those
zoom
had the
out,
little
I'm gonna
white,
have you about 8.5 in my
I don't know if they're gametes along the
And ship is
center
still
of
moving.
the...
Okay. Video, could you pull in so I can see the skids very briefly?
You're full wide.
Oh, full wide?
You are
Okay.
full wide.
Okay, great, thank you.
Yes, you might just want to climb a little bit.
Yep, I'm heading up now, co-pilot.
Getting into some pretty steep slope here.
And we're hearing from Nolan Barrett, who has reviewed the sponges we've seen, and he's saying that some of the ones we've seen are true geodias, a couple of small Afrocalistes, probably A. beatrix,
Absolutely.
and some
watch lead.
round, smooth demo sponges that he is not certain of
It's
their
going to have to be
species.
a brief look.
Yeah, I'll
There's
keep
a
an eye
crab on the
Should be
right.
going to
too.
it.
Yeah, I don't know if that was a Chaseon crab.
I don't
We
think
didn't-
it was.
But this is
Chaseon?
one of our first soft corals that we've seen, so I thought it'd be worth a look.
Oh yeah, definitely. It's beautiful.
And you're interested in the coral here, watch lead, to the left of the lasers?
Yep, roger that.
Okay.
Isn't that pretty?
Yeah, you'll be coming to an end very shortly here. So
Copy.
if you need to turn and look back at him.
Wow. This is a nice cluster. At
we
least
wanna
3, 4.
Perform some zooms on this, watch lead.
Yes, definitely.
So I should sit down for a zoom.
Oh, yes, sorry. You can see one of the Venus flytrap on the right of the cluster. There are three that are wide open, and then to the right is one that's closed in, so you can see what they look like.
Looks like a shark.
Giant mouth. They look threatening even when they're closed.
I was thinking it was Anthothela, and that's what Tina also is thinking, although the zoom will help.
This is a soft coral. Now that's a pretty scene. I don't know if I'd call the one on the upper right pretty, but that's beautiful right there. I'll bet that ends up in one of the screenshots on the web. That's beautiful.
Copy that, Bridge. Thank you.
Is there anything in particular that we wanted to look at?
Now, if you could pan down.
Co-pilot, would you grab it?
I think there's another of the Bathanectes.
Sorry, say it again.
Would you
crabs.
grab the Zeus?
Zeus? Yeah.
And rotate down?
Yep, I have your Zeus and I'm tilting down.
Copy.
Is that what we
I guess
want?
tilt down is the proper term.
Do we
Panning
keep going?
is right and left.
Left and right, yeah.
It's probably not into the view unless you zoom out.
Is it further down? Watch leads. Just gonna keep going down, pilot, until we hear something.
Is it up or down?
It's
or
down. It's farther down. It was
Okay.
more in the foreground.
There he is.
Yeah, right there.
Yeah, that looks like a Bathanectes.
Yep.
Those are swimming crabs. You can see their last pair of appendages have kind of a wider, I guess you could call it kind of a paddle.
And Bathanectes means deep swimmer, doesn't it?
Yeah, I think
Yep.
so.
That's great. Thank you.
Right. Full wide video?
All right.
And would you? Great, thanks.
Yep. Move your camera.
Okay.
And you can see another Bathanectes
Then
up
I'm
and to the
still,
left.
But I'm also looking.
Then
as
there was
far
another
back
crab on
as
the
I
right.
can.
We'll see if we can
Okay.
see
I'm
him
gonna
as we
pick
fly
up.
away.
And make some speed.
He's just off the view on the right. Just got a glimpse of him.
Need?
Huh?
Never mind.
A fair amount of that Anthothela octocoral and lots of anemones around here. Some more of that octocoral.
Yeah, right now, watch lead. We're kind of a little bit behind Sirius and the
Okay.
ship, so we're gonna.
Yeah, we figured.
Yeah, we've been stopping and zooming so much that the ship is now a little bit far away, so we're going to make some time and catch up a little bit. And we're now at 387 meters. Lots of anemones.
All right. It's about 80 in my camera now. So we're starting to get in front.
Okay.
And still looking about 135, 140.
It really looks like a garden of anemones,
Yeah.
and they're all in bloom. Just as we're passing over it, we're kind of leaving that area, but it was interesting how clustered they were there.
Mm-hmm. Conditions must be perfect for anemones
The
there.
metropolis. We're moving out into the suburbs. Actually, as the lights hit them, we're still seeing just as many.
Mm-hmm.
Copy.
So I see a good return up there in the distance, 37.
Oh, it looks like it might be
Wow,
another
look at the
dip.
current.
Ridge, yeah.
Loved it. Yep.
So we are heading in a northwest direction.
Yeah, definitely a lot more than before.
Oh, no, I'm sorry. I have it backwards. We're in a southeastern direction. The opposite. I got the back from the front mixed up on the view.
We're caught up again with the ship. Watch these.
Great, thank you. Sounds like we're caught up with the ship, so we can have more maneuverability and zooming. If anybody sees anything that they want to take a closer look at, let us know.
Like.
Yeah, I think it's fine.
Copy. Yeah, sounds good to me.
Okay, maybe 135 or so.
Yeah, sounds
Kind of
great.
what the
There's another
that's happy.
crab on the
Oh, yeah.
just next to the lasers now.
bridge, RV nav.
So that looks more like cancer.
Yeah,
Genus
Hey, we're ready
cancer.
for another
that's
move. 30 meters at 135 degrees, 0.2 knots. Good copy.
and that's what we saw earlier. Just didn't get a chance to take a peek at it.
Can come in for a partial.
Lasers are clear.
He's small.
Lasers are
Remember
coming
that
out.
those lasers are only 10 centimeters. So his carapace is only about 10 centimeters in width.
It's a little tiny sea star just
Oh, yeah.
behind the crab. You can see that these crabs have very stout claws, and they can break into crustaceans or bivalves.
Copy
Or
that.
Little tilt up.
Stand by.
It's a cool shot.
And one of the anemones just closed up. Maybe it caught something.
either that or got trampled.
Okay, full wide.
Wow, look at all those anemones.
Pushing past the skates.
Okay, so I'm going to let the current bring me over to port.
So we have
Copy,
flytrap
it's
anemones, the big one in the middle and just to the left, and then we have the smaller orange anemones that have been carpeting everything around here, and then there's the white anemones with an orange mouth, the center. Quite a beautiful scene.
It really is. Those pinkish ball things you're seeing in the foreground, those are the Venus flytrap anemones just closed up tight.
Our thrusters probably throw a little bit of water movement at them, and
Hoping that we'll hear from our sponge person to get an ID on these sponges that we've seen 2 or 3 of right in this area. Nolan Barrett, if you're listening. Looks like there's something of interest on the left of the screen.
Pilot, do you want
Or is
to
that just anemones growing
Oh,
on
yes, please.
copy? Lasers coming on.
a raised piece of
I
coral
can't
skeleton?
tell what that is.
Yeah.
Pilot,
We
if you
take
want
a
to. It's been about
moderate
half
zoom
time.
just above your
You still want
lasers.
to change out or swap.
I guess it is a cluster. It looked striped, but it is just
Okay,
the
now.
stalks of an. That's
Just-
all right. I think we're okay. Now we see
Okay.
what it is.
All right.
We both thought it was some fish or something with stripes.
Okay, I'm setting up now.
And we're at 380
Let me just
meters.
see if it's.
And the top of this feature was supposed to be about 375, so we're getting close. Gonna just climb up a little bit further.
That was the quickest pilot change I've ever seen.
Musical chairs.
We're quick.
Have we had a front row introduction of late since the latest shift?
I don't think so.
Not at all.
Okay, I guess I'll go ahead and start. Sitting in pilot right now.
Okay, yeah, sorry.
Do you want to talk on?
Okay, yeah.
Okay. Sitting in pilot right now is Andy Lister, and over in co-pilot.
Co-pilot is Sean Kennison.
And ROV navigator is Andy O'Brien.
In the far right corner is video is Roland Bryan.
Oh, and in the back row is Annie White on clipping, and we're all with the Global Foundation for Ocean Exploration.
Current picked up a little bit, huh?
And kudos to all.
There's like weird
And you
Bridge
guys
now.
are?
ridges.
Yeah, I can see the.
I'm
sonar
Leslie
up ahead.
Sauter from the College of Charleston Geology
I'm gonna go lower
Department,
this so we can see
and
Great.
I'm
That's
the
what I was just
Geology
calling to
Watch
ask
Lead.
about. Thank you, Bridge.
And I'm Cheryl Morrison from the U.S. Geological Survey Leetown Science Center, and I'm the Biology Watch Lead.
And we are all part of the Okeanos Exploration Program.
So let you get a little bit farther out, pilot, and then put another move in.
Sounds great.
Just pushing past the pull string.
Sounds good. Does it look a little blurry? Yeah. So watch as if you have anything in this area you'd like. Feel free to shout out.
Pilot, right behind this sponge on the right side, there's one of these feathery either corals or hydroids that we've been trying to collect. I don't know if this might be a possible place to give that another shot, but if not, we'll see many more. So
Are we? Is it? That's not the one that's in the laser. Oh, it's over here to the right.
yes,
Okay.
just behind the orange pinkish urchin.
Urchin.
I was trying to see if
I
that
know.
was a closed-up anemone. We've seen so many anemones.
And maybe this one is low enough and on the sand that a scoop would work.
That could be. A scoop might actually
If we
work
come
here.
at it from the other side.
What do you think, co-pilot?
We've been shooting for this.
Let me get settled and we'll zoom in and see.
Okay.
I think it's still on the crumbly bits, but
Oh,
still think
surely it's on the
Alright, let's
crumbly
go.
dead, but
Just
maybe
snip it and
underneath
grab
that
it.
is sediment.
Yeah, maybe we could grab the
Scoop
whole
it all.
crumbly bit as
Yep.
well as some sediment with a big scoop. And
so the
And
only
then-
thing I'm afraid about here is the current has picked up, and
okay.
it might just take her right out of that scoop.
Yeah, we were hoping that if we had some of that skeleton rubble, that it might weight the whole sample down. But
It's tough to say.
Which is the real
That is
reason
very
for the
tough to
scoop.
say, but
and It looks like an urchin test
shell
on the
test.
right
You want to
side.
zoom into it?
Yeah, just give me a second. Okay, go ahead, zoom in. And let's get lasers off.
Yeah. Lasers coming off.
The ship's stationary.
What do you think, co-pilot? You think a scoop would... I know I can get it without the scoop, but we won't have the coral underneath it.
I still think that, I mean, the best method would be to snip and grab with the tag on.
I think so too.
But if they want...
So watch leads, we can try with the scoop. We think that the best method is definitely going to be grabbing it with the tag on and getting the whole sample in the box.
If you want so after that, you can
But
go back with the scoop.
yeah, we could always go back afterwards and get
Right,
some
right.
soil.
That's, yeah.
Yeah, that works. That works fine.
You know best.
Okay.
What if you were to use the tag on upside down so you actually cut the top off and have the tag on the bottom?
So you want to go ahead and spin up? It's already spin up.
I guess if it doesn't float away after the first attempt, then it's attached
pull
to
up.
something. So
Yeah.
Can I
that's
take
good.
your screen?
Yes.
Okay.
And pilot, just so that you know, this is likely to be very floaty.
Okay.
So
All right. So yeah, I think I'm just going to go straight into my port side and get it in there real quick.
great.
All right, port. It's ready. Do you want me to lead you with...
Yeah,
lasers,
I
or-
can get out there. I'll get out there with it, but we might zoom in a little bit. Okay, coming on. How's it been behaving today?
Fine, no issues.
Okay.
So
Great.
we've been debating whether this is a black coral or a hydroid for this whole dive, which is why we wanted to collect one. But it looks
Almost
like there's more of a
too close.
consensus that
Might
it
have
might
to-
be a hydroid now. Even if it is a
Yeah, it's about
hydroid,
to
it
snap.
has an
you're
unusual
right.
form.
So you could just, if you rotate it a little, or just backed up a little, or rotated starboard.
With D2, or are you talking about the arm?
I bet with D2, but that might work too, what you're doing.
I was just going to come at it from this side.
That looks great.
Oops. Got a little extra for you.
That'll make another sample. That's fine.
Hey, the green is grip lock, right? It's not there.
Right there.
Yeah, I need to hit it.
It's not lighting up?
It will now.
Oh, you have
There
to
you
be
go.
outside of... Yeah, you
Yep.
Next.
Okay.
All right, let me know when you're ready for a drawer.
Is that? Do we need any zooms or anything here, or you just want to get it in?
That's fine.
Okay.
We can just get it in since it's so floaty. I'd hate to lose it again.
Okay. Yeah, go ahead, come out.
All right, coming out. Oh, the one's actually still there.
Oh yeah, we actually do have the other sample. Still,
really?
It looks like if you look on our
Barely.
right screen up there.
We can't see it in this.
Oh yeah, it's out of the way
It's in the,
right
uh,
now.
Oh.
See in the top right little monitor.
I see.
Yeah.
Look on your multi-view, it's on your bottom right.
Yeah.
Do we need to
So
tilt
our
down
first
with
sample
lasers?
actually might
go ahead.
come
I'm
with
actually
this
using
to
the
the
nip cams,
surface. Is that on the edge
Alrighty.
of the rock box, I guess, that it's caught?
That's actually the right corner of the sample box,
Okay.
bio box.
Oh,
Okay.
so it was escaping.
Yeah, we thought it all the way escaped, but
What do
well,
you think?
that's
Is that a
good.
good depth there?
Yeah.
I think
Just,
so too.
Yeah.
It does make
It
it
does
sample
make
too.
it number 2, yes.
My downfall was coming out too quick, and I kind of
Copy.
moved
Sure.
the water around.
Okay, great.
Yeah, it's sinking. All right, I'm going to close?
Go ahead.
Great. Nice job.
Got him.
So as far as the scoop, do you still want to try a scoop for sediment, or not too interested, or whatever you guys would like?
I'm sure that Leslie's interested. I'm a little bit worried.
We don't
about
want to reopen
moving,
that box.
Yeah, moving
right
either
now.
box now.
Yeah. We'll keep moving. There's more sediment ahead.
Copy that. If you want to follow me back around for this, for the stowage.
Yeah.
That was Port Outer. Yeah.
Nicely done, Pat.
Thank you for the assistance there.
Now, do you want to write down too that there's a specimen caught in the starboard lid? If it opens, it's probably most likely going to float away. But we have the same sample in the
port outer.
So
I think now, once that it's in there, I think we can probably safely open that slowly and put another sample in there. But
that would weight it down for future collections.
That's true.
Yeah.
As long as we're slow about opening and closing it. Because when I opened the jaws, it did seem to settle.
I could go either way.
That's good.
What do you think?
But if you, as you dump though,
I could.
the mass
could easily
just float it out.
Well, we could definitely put a sample in the port inner, I think, if we're careful.
Yeah, that's great. Let's do that.
Go.
A scoop sample.
Yeah, that'd be great. And we can get the rubble, which several people have requested.
Oh, do you want to do it now or in a little bit?
While we're here, before we stir it up again.
Okay.
Yeah, and also there aren't that many places where there's exposed sediment that we've seen so far.
This is a good sediment and rubble and organisms living in the rubble. This is a good spot.
Copy that. Hey, do you have any tips, co-pilot, on the scoop?
Ooh, that
try to grab it like
Umuta Picta just tried to get that coral hake
back a little bit.
in the upper left-hand
like
corner.
Oh, really? Oh, look at it.
Don't grab the scoop straight on.
Yeah, you want a little bit of an angle. Gives you a little better scoop angle.
Watch leads, Nav.
And we use main HD and we put the wing inboard a
Go
little
ahead.
bit.
Okay.
Do you want me to do that?
Yes, please. I haven't used the
Starboard
scoop
wing
too much
inboard.
at all.
Watch lead, Nav.
It's usually about right there. And
I just wanted to confirm that spec 01
Yeah, go ahead.
bio.
So I'm going to come out from it and kind of from the other side of it, yes?
it doesn't have to be extreme. Just almost
Copy.
like that.
Thank you.
And just, angle back a little bit. And like that. You should be good. Yeah, something like that looks great.
Okay.
Use your mini Zeus though and your minute. See your little offset.
More like that, you're saying?
Yeah. Yeah. And just close it slow and the fingers will find it. And you know how to increase grip force. I don't know what
I
you're
did.
at.
I put it at 5.
Okay,
I
great.
think that's enough.
Yeah, should be
Okay.
good. I think you're slightly off. Yeah.
There we go. That's
That looks
better.
great. Yeah.
Okay.
Lock it in. Just pull it out.
What kind of angle do you normally use?
Like that angle.
Okay.
I don't know how tough the sediment is, but
Huh. Not
Maybe try to
exactly not tough.
maybe try to straighten it a little bit. So you're kind of almost hitting it with a corner.
Well, I got a lot of other stuff.
That's great. That's what we want. Substrate.
Looks Looks good to me. And then let me know when you're ready, and I'll come out slow with the drawer. And then you might want to straighten up before we just your arm up a little bit, make your life a little easier when you dump it.
Current is starting to blow it.
That's right, you still have plenty.
Okay, let's start from there, and I can straighten as needed.
Copy. Sending drawer.
Okay. That's the way they work. Okay, keep an eye on the floaty, even though I'm not going to go near him.
And then you could just,
like that? That's what I was thinking about, just pouring it in and coming up a little bit.
No, you're doing great.
Okay.
You might have to shake it a little bit, but yeah, see that?
Might get a little bit deeper in there, like that.
Yeah.
Oh, that's one getting nice and dirty.
That's what we like.
So
Some of us.
That.
I think that's pretty good there. So I'm going to come on out.
Close the drawer.
Yep. I could try. I don't know that the scoop's going to be the way to go, but let me actually confer with co-pilot. What do you think on scooping the urchin or lightly? I think I'm going to crush it with the claws, though.
It's small, too.
It is small. I could try real quick with the scoop.
Yeah.
Okay.
While it's out.
Okay, let's get this guy away. Get out of
You
here.
could put it in the same box as the sediment.
We can certainly try real quick.
Okay, it could go in the same box as the sediment. It won't matter because it's not alive anymore. And
Okay. Sorry about that.
no problem.
That's all right. And it was living on the sediment anyway, so that'll be okay in terms of associates.
I think it was these two. One of them.
Yes.
That's actually the same one that's in a couple pieces, I think. So.
And for anyone annotating, that sample we just collected is
watch me closely, 'cause this is gonna be a weird move.
03 Geo.
Yeah, now you look good.
This will be 04 Bio if it's successful.
Nice.
Ooh, nice job.
Come on, don't blow away. That's what I was afraid of.
If you push it up against, see all the other dead coral?
Yeah, let me let it clear real quick so I can see it.
Yeah.
Yeah, I got you. So just kind of come in with it.
I'll just do the same thing, and then once it hits the dead coral, it might help push it in.
Okay. Here we go. Round 2.
Do you want me to center up Mini Zeus?
Yeah, please.
Yeah. I think it's in there.
Oh, please don't blow out.
Yeah. Alright, ready for extend?
Ready.
Coming back out, slow. Can I tilt Mini Zeus down?
Yes, please.
Alright.
Okay, first box. Here we go.
It's all the way out.
Okay, watch me